Hello and welcome to the course Sparse Modeling of High-Dimensional Biomedical Data, which is the new course for the summer semester 2025. My name is Christian Staerk, and I have been a jointly appointed junior professor since April 2024 at the Leibniz Research Institute for Environmental Medicine, IUF, in Düsseldorf, and TU Dortmund University. And this course in the summer semester will be at Dortmund University. Now, what is this course about? It's about sparse modeling of high-dimensional biomedical data. And nowadays, we often and more collect these high-dimensional data sets. So, for example, we have data on the so-called exposome, which encompasses all kinds of environmental factors like air pollution, sun radiation, nutrition, temperature, stress, and so on. So you can imagine there are many different environmental factors. And then we have also the genome. Okay, so here you can see two different individuals with their genome. And actually, most of our genomes are the same. So 99% of our genomes are the same. But if we look at the positions where we might differ, so-called SNPs or single nucleotide polymorphisms, there are still quite a few of these positions where we might differ. So 10 millions of them, 10 millions of SNPs. And even if we go one step further, look at the level of genes, which are parts of our DNA, we still have 20,000 genes in humans. So we have many variables. You probably also have clinical variables, socio-demographic variables, and so on. And taken together, you have many different variables P, while your sample size N is oftentimes much smaller than the number of variables P. So the number of people in your study is relatively small in comparison to the number of variables you have for each individual. So in order to deal with these high-dimensional data sets, we really need targeted statistical tools. We also need scalable algorithms, so computing is important. And we, of course, have to keep in mind what is the biomedical question we want to address. Now, where does sparse modeling come into play? Now, if we have so high dimensional data, we have to assume something, right? And the sparsity assumption is quite natural. So it just says that, in fact, out of these many variables P we collect, maybe only S, where S is much smaller than P variables, are important. So instead of 10 million variables we measure, maybe out of these only 10 or 100 are important. And so we want to use our data to do the variable selection, right? So the aim is we want to identify the important variables, for example, genetic and environmental factors, let's say for skin aging. And also we want to develop prediction models to say, okay, what is the probability of, let's say, to get skin cancer? Now, what are methods we will cover? So this is just a brief glimpse into the methods we will consider. So this is a very broad class of methods: regularized estimation. So here, imagine we have a vector of regression coefficients beta, which we want to estimate. So each component of beta, let's say, corresponds to one variable. And we want to estimate the effect of this variable on some response variable, some outcome variable. So we have beta in RP. And now we have observed some data and want to minimize the loss. Okay, so this could be the least squares loss leading to ordinary least squares estimation. But if we have high dimensional data and just focus on minimizing this loss function, we will just overfit our data. So if the number of variables P is larger than N, the sample size, then this would not be very informative. So usually we need some kind of regularization. And this is the second part here. So we have lambda times the R beta here. So lambda is a tuning parameter and R beta is a regularizer or penalty in dependence of beta. And depending on how we choose this R beta, we get prominent special cases. So if we choose R beta to be the L0 penalty, which is not exactly a norm, but it just counts how many coefficients are non-zero. then this would lead to best subset selection. So this is the most natural thing, right? We just count how many variables are inside our model and we directly penalize for that. So this leads to sparsity. So I would say it's really the most natural thing to do to have sparse estimators. But the problem is this leads to discrete and combinatorial optimization problems, which are very hard and computationally challenging to solve for large P. So now what is usually done nowadays is something like the lasso or variants of the lasso, where we replace the L0 penalty with an L1 penalty, which is just the sum of absolute coefficients. And you can show that you will also end up with sparse solutions. This is nice for interpretability, so you get variable selection. And at the same time, it's a convex function, right? So this L1 penalty is convex. And if the loss function is also convex, then we have a convex function we minimize. So you can use efficient algorithms to solve it and compute the estimator. Now, we can also go one step further, right? So we can use also an L2 penalty, which is often squared for convenience. But this is very simple to compute in a linear regression format. So this will will have a closed form solution. But the problem is that does not directly lead to variable selection because in this optimal solution, there will be usually no exact zeros. So there will be no variable selection. So all variables will be somehow included in the model. So this was just a very brief overview of regularization and variable selection. So we will go into detail in our course, which will be an important part of our whole course. The first part on lasso and many variants like elastic net, adaptive lasso, and there are also other more targeted penalties like mixtures also of which go in the direction of L0 and L1. Can we go a bit more towards L0 penalties? Then we have ensemble methods where we apply different methods or the same method multiple times on subsamples of the data to get stable variable selection as in stability selection. Then the whole field of selective inference and uncertainty quantification is actually a hot research topic currently because if we use our data to select the model, select the variables, and use the same data again to do selective inference, so we want to compute p-values and confidence intervals, this is actually not straightforward, right? So standard p-values and confidence intervals will not be valid. So this is something we will highlight and also discuss some solutions in that context. Then you have probably heard about interpretable machine learning. So there you have more flexible methods like boosting and random forests. And you still want to understand what are the important variables for the prediction in these more flexible machine learning approaches. And the last point is on sparse graphical models, which is now unsupervised learning. So the first four topics here I discussed will be on supervised learning, where you have variables and one outcome variable you want to predict. And in the last part, unsupervised learning, you just want to understand the relationships between variables. Let's say you have genes and you want to estimate conditional independencies between the genes to have a gene network. And for this, we will use also tools from graph theory to get sparse graphical models. Now, in all of these methods, we will consider statistical properties and computational properties of the methods, and also apply the methods on high-dimensional biomedical data. So just to sum up some organizational stuff, the target group for this course is Master of Science Statistics, Master of Science Data Science, and also interested graduate students. We assume that you have basic knowledge in linear algebra, calculus, statistical modeling, and R. The structure of the course will be 2 hours lecture per week, and every second week we also have a tutorial. These are already the timings. The tutorial will include both theoretical and practical parts using R. We will decide about a written or exam depending on the number of enrolled students. And here's already some literature. So very nice books. The first one by Hastie, Tipshirani, and Wainwright on statistical learning with sparsity, the lasso and generalizations. And the second one by Lederer, Fundamentals of High-Dimensional Statistics. It includes also very nice exercises and R-labs. So if you are interested, you can already check them out. And finally, if you want to explore the world of high-dimensional biomedical data, I'm very looking forward to welcome you in the course. So please consider joining the course. My name is Christian Staerk again. And if you have any questions, please feel free to reach out. This is my email address. I'm looking forward to seeing you next summer semester.